Share the screen. All right. Oops. When did that go? Oh, that was my Zoom for the lesson. The lecture just finished. Let's come back here to where I was. So we're going to be talking about Lab 5 today, streaking for isolation and culture media. Also read the unknown projects file, the instructions on Canvas for next week. I'm going to talk about the unknown project next week. And it looks like, am I sharing my screen? Anyone confirm that? Oh yeah, I am said stop share, okay. It looks like next week I'm going to discuss the unknown project on Tuesday, and then we'll have Lab 6 on Thursday. So we will be meeting both Tuesday and Thursday next week. So you need to read the unknown projects file. Let me see if I can find it. It's not in. I think you can find it under week 4 or week 5 of the lab. No, I just have the warning, read about it in week 4. So in week 5, there it is, the unknown instructions. So you should read that before I begin talking about it next week. I'll also talk about the others in the unknown report, the sample journal, the building autonomous game plan, and the unknown list and the unknown notes, but you don't need to read those before I talk about them. But it would be a good idea to read the instructions. All right, so let's talk about Lab 5. The cultivation of bacteria, culture media, inoculation, and isolation. There is some reading to do in the textbook, so please do that before beginning the lab. There are some video clips and a virtual lab today. The primary learning objectives, upon successful completion of this labs, you should be able to understand the importance of proper specimen collection and transport to the laboratory, explain the 5 principal steps used by microbiologists to process specimens, be able to differentiate between various types of culture media, and explain the purpose of street plate culture technique. And this is more commonly called streaking for colony isolation. So when you streak for colonies, it's a streak plate culture technique. Be able to define the following terms, which is selective media, differential media, and reducing media. So we'll talk about these different media types today. So be able to isolate a pathogen from a clinical specimen submitted for culture. And when you do this, it's only as good as the collection technique and the timely transport of the specimen to the lab. Too much time, the bacteria can die. And if you don't use good technique, you can get your specimen contaminated. And if the specimen is rare, getting the contamination in there may make it impossible for the lab to isolate what's making this patient sick. Some microbes are sensitive to desiccation, that's drying out, or exposure to oxygen, like anaerobes can't be exposed to oxygen, and therefore they require a special transport medium. Once a specimen is received in the lab for microbial culture, microbiologists employ the 5 principal I steps to process it. First, there's inoculation, then there's isolation, then there's incubation, there's next inspection, And lastly, identification of the specimen. Inoculation is just placing the specimen on an artificial culture medium to begin its growth. Isolation would be spatially separating out the microbial growth into distinct colonies. And if you have more than one species there, get them isolated. Incubation, placing the culture media in the environment under conditions to facilitate their growth. And generally, we warm things up, bacteria up to grow them. There are some exceptions, obviously. Inspection, examining the culture for the presence of microbial growth, and then identification, trying to identify the species of bacteria you have growing, assuming you've got it isolated into different species, different colonies, and usually performing specific tests to identify any pathogenic microbes that are isolated. These 5 tests use different types of culture media. And so let's talk about the different types of media. There's selective, differential, and reducing culture media, as well as general purpose media. And we've already talked about general purpose media. NA, NA or NAYE, and TSA are all general purpose media. These media grow many organisms and they don't select for anything. They don't grow all organisms, but they're cheap and they grow many organisms. Organisms that would not grow on a general purpose media, we call fastidious. To promote the growth of fastidious microbes, we need to add something to the media to get them to grow. And these we call enriched general purpose media. For example, blood agar adds 5% sheep's blood to the media to help an organism grow. For example, Streptococcus pyogenes, the bacteria that causes strep throat, will not grow on most media. You need an enriched general purpose media like blood agar to grow Streptococcus pyogenes. Because this bacteria is fastidious, it requires certain additives to the media to get them to grow. And a cheap way to add a lot of things is just add red blood cells. And then the red blood cells will supply the nutrients that a fastidious organism, well, I should say Streptococcus pyogenes, requires to grow. Different fastidious organisms have different requirements. Any question about that? All right. So besides general purpose media and enriched general purpose media, we have selective media. Selective media has chemicals in it that inhibit or halt the growth of some organisms, but allow other organisms to grow. Keep in mind, if you're using selective media, first, the selective media isn't necessarily perfectly selective, meaning it may not be 100% selective. You may get growth, but it will be reduced in its growth rate. So it's selected against, but not 100% selected against. And second, even if the organism is supposed to grow on the selective media, it may form smaller colonies or grow more slowly than it would on a non-selective media. All right, any questions about selective media? There's also differential media. It too contains chemicals, but these chemicals don't select for different things to grow. They allow us to see visible differences among different cells, different species. These differences are unrelated to how well the organism will grow on the media. Usually you will have a different color between 2 different species, but the difference is they will look different. and that's the differential media. Differential culture media allow dissimilar bacteria to visually be distinguished from each other. And we'll tell you about some examples in a little bit. Lastly, the last media we need to talk about is a reducing media. A reducing media promotes the growth of anaerobic bacteria because anaerobes require low or no oxygen conditions in order to grow. Now it's tricky getting these to grow if they're strictly anaerobes because any exposure to a strict anaerobe, any exposure to oxygen will kill the cells. So you have to keep them totally in the media. It's important to note that a culture medium can be classified in more than one category. For example, McConkey's agar is both selective and differential. And blood agar, which I already mentioned, is both an enriched general purpose media and it is also a differential media. Any questions about that? All right, here we're saying McConkey's agar. It is selective in that gram negatives can grow on McConkey's agar, meaning it has chemicals in it that prevent the growth of gram positive organisms. It also has the sugar lactose in it and a pH indicator. And so that will allow you to distinguish the lactose fermenting bacteria, which will change it to a pink color from the acids building up from fermenting lactose, from the bacteria that cannot ferment lactose, and they will not be pink in color. So McConkey's agar is both selective and differential. Like I said, it does not grow gram positives. Both of these are gram negatives. And it's differential that we can distinguish the lactose fermenters from the non-fermenters. Any question about that? We've already talked about blood agar, and that's an enriched media, and the blood supplies the nutrients in this media. For example, Streptococcus pyogenes will not grow in most media, but it will grow in blood agar. So blood agar is an enriched media. It is also different, excuse me, it is also a differential media because you can see differences in the hemolytic patterns of the bacteria growing on the blood agar. You can have bacteria which do not perform hemolysis of the blood, and that we call either not hemolytic or gamma hemolysis. No hemolysis. And you can tell that because looking at the blood, it's red. It's red all the way up to where we see the white of the Greek letter gamma. That's what that stands for. There's no hemolysis. Any question about that? There's also beta hemolysis, and that's complete hemolysis. When we're looking at the blood here, it's red here, and then it becomes dark, and then it becomes clear, and then the white here in the beta, that's the bacteria growing. This part there is actually clear, that's not white. You can actually see through this part of the agar. And this is complete hemolysis or beta hemolysis. Any question about that? The third pattern we can see is partial hemolysis. And when we see partial hemolysis, that's called alpha hemolysis, we notice the blood, and it's a very light ring, very skinny ring of partial hemolysis, but there's this dark ring there, and then there's the white of the bacteria, or off-white, you might say. And then you see inside the alpha there, it's darker. That's from the partial hemolysis. This partial hemolysis is darker. Sometimes it takes on a greenish appearance. And that's from some of the blood being lysed in the partial hemolysis. So out here, no hemolysis. Out there, a partial hemolysis. And then that's the bacteria there. You'll notice whenever we have beta hemolysis, the clear zone, around the beta hemolysis, we'll have a ring of darkening, and that's alpha hemolysis, where further out we have partial hemolysis. And that's something you normally see. All right, any question about blood agar and hemolysis? All right, so this table is showing you the different types of media and talking about them. The general purpose media, TSA, NA, NA. The differential and enriched media, there are really only one we're going to talk about, and that's blood agar or BAP. And that's TSA generally with 5% sheep's blood added. And this is both enriched because of the blood supplying nutrients and differential. We can see the hemolytic pattern differences in the blood. Chocolate agar or CAP is only an enriched media. It is the same as BAP, meaning blood agar. It's just that we add the blood when the agar is still quite hot, right out of the autoclave, and that lyses the red blood cells. And that gives the color a brown color, and it's called chocolate just because it's brown. Chocolate agar because it's brown. Okay, then there's selective and differential media, and we have MAC or McConkey's, EMB or eosine methylene blue, and HE or hectone agar. They're both selective and differential, meaning they have chemicals in them that allow certain bacteria to grow and do not allow other bacteria to grow. That's why it's selective. And all three of these that we're discussing here allow gram-negative bacteria to grow and select against the gram-positives. It's also differential, and all three of them are differential on the basis of lactose fermentation. For McConkey's, as I mentioned, if the bacteria ferment lactose, that sugar, then they will be pink in color. If they can't ferment lactose, they'll be non-pink. The others will be a different color, like hectone agar. I believe that one is salmon colored if it ferments lactose. PEA, or phenyl ethyl alcohol, is only selective. It selects against gram-negative cells, and we use it to help isolate gram-positive bacteria. Mannitol salt agar is also selective and differential. It has chemicals in it that prevent the growth of organisms that do not grow in salt, and the only chemical is actually sodium chloride, 7%. And if it grows in 7%, it'll grow on this plate. If it doesn't grow in 7% sodium chloride, it won't grow on this plate. So that's selective. It's also differential in that it allows you to distinguish between bacteria that can ferment the sugar mannitol. If they ferment the sugar mannitol, they will have a yellow ring around them. And if they don't ferment the sugar mannitol, they will be the normal color of the mannitol salt plate, which is a pinkish purple color. All right, any question about this table? You'll need this table to answer the questions in the worksheet. All right, so the 5 principal steps: inoculation, that's taking the growth you get from a patient and inoculating it on the appropriate media. Isolation is isolating out the different species of the bacteria you have, and then growing them up as a pure culture. This concept was first developed by Louis Pasteur in the 1850s, and he came up with a way to get a pure culture of one bacteria species. His way of doing it was not very efficient. He used limiting dilution. I'll talk more about that in the lab. It was Robert Koch who developed an easy way of getting a pure culture. And that is, you take out the bacteria and you streak it on a solid culture, like a solid agar plate, and then get isolated colonies to grow. If you get an isolated colony to grow, you take from that colony, streak it out on another plate for isolated colonies. And if you go through 2 isolated colonies before you set up your bacteria to grow it up, then we say with a good chance you have a pure culture. And the chance will be greater than 95% that you have a pure culture. If you only do it one time, your chance of having a pure culture is something like around 75%, meaning occasionally a colony can be formed by more than one cell. And if 2 cells form the colony, it may not be one species. It could be 2 different species. All right, any question about any of that? So let's talk about ways you can get pure cultures. Theoretically, the quadrant should have individual bacterial cells from isolated colonies after incubation. What you do is you get your bacteria on your loop and then do streak 1, quadrant 1, right there. And it'll have a whole lot of bacteria cells in it. And then you sterilize the loop, and then you do streak 2 or quadrant 2, and you streak it out on the plate. You actually shouldn't come all the way down to here. You should only do, maybe come this far to do quadrant 2. The reason is there will be more bacteria here and less bacteria here. And so if you come and set this quadrant up, you have a sterile loop, and then you put it into here, you'll pick up bacteria from these streaks and then set up quadrant 2. You come all the way here, you'll get the highest concentration of bacteria cells, and you want less concentrated bacteria cells. And these last 2 loops will be more diluted than the first 2. And ideally, you want to go 20 to 30 times in a quadrant. You don't want to do 1, 2, 3, 4, 5 or 6 times in a quadrant. You want to go 20 or 30 times, 20 to 30 streaks in a quadrant. Any question about that? And then when you set up quadrant 2, like I said, you do in the more diluted portion of quadrant 1. You don't go into the high concentration of quadrant 1 to set up quadrant 2. And then you set up quadrant 2, and then you sterilize your loop. And then you, once again, this isn't quite done the way I would do it. You set up quadrant 3 by streaking it into the lower portion of quadrant 2, meaning the more diluted portion. And you go into quadrant 2 3 or 4 times to make sure you pick up cells, and then streak out quadrant 2. And actually, it looks like they did quadrant 4 correct. And that is, it's not going into the high density areas of quadrant 3. It's going into the lower density areas of quadrant 3. And then set up quadrant 4. And the goal of having these different dilutions of cells, there will be more here and then less here. And then this will be much more diluted than here, because we're starting with nothing and then streaking it into quadrant 1 to set up quadrant 2. And the idea of these dilutions is somewhere along the line, we'll be plating out a single bacteria. Like if we plate out a single bacteria right here, it'll then be allowed to grow up into a colony, and that'll be an isolated colony. When you do your colony streaking, you should have at least 4 or 5 isolated colonies. That's a colony not touching another colony. And gosh, there's a large number there. I would say at least 10, maybe 20 or even 30 isolated colonies there. You need 4 or 5. Of course, we can't do this in real life, but if you were in a lab, I would require you to get 4 or 5 isolated colonies. And all you need to do is practice, and then you can do this. Any questions on any of that? You do not need to get the isolated colonies here in the 4th quadrant. They could be in the 3rd quadrant or even the 2nd quadrant. Very rarely would you get them in the 1st quadrant. To get them in the 1st quadrant, that would mean that the bacteria that you're putting on the plate is very diluted to begin with. So it doesn't matter where the isolated colonies are. The goal of this quadrant streaking method is just to get isolated colonies someplace on the plate. Now remember, before we call it a pure culture, you take from this isolated colony and you streak it out in the 4 quadrants again for more isolated colonies. And only when you pull from cells that have been gone through 2 isolated colony events do you call it a pure culture. And even then, it's not 100% pure, but it's over 95% of the cases will be a pure culture. All right, incubation is just taking the cells and then putting them in an appropriate environment for growing. We call that an incubator. Fancy incubators can control the oxygen, the carbon dioxide, the temperature, the humidity, and maybe a few other things. Our incubators in the microbiology lab, if you were really attending the lab, all they do is control the temperature and the humidity. All right, any question about that? You then inspect the growth and look for observable visible colonies, and then note the differences between the different colonies. Usually, if you have a mixed culture, you can see differences in the colonies. They can vary by size, shape, margin, elevation, and Lab 1 can review those terms. And then last, you identify the pure culture. So if you have a species you think is causing the patient to be ill, you then run appropriate microbiology tests to try and identify that species. And these are the biochemical tests that we'll begin to talk about. All right, any question about any of that? So know the terms of this lab. So there's a little video you can watch on inoculating agar plate using the streak plate culture method. You only need to watch that to 4 minutes and 15 seconds. Note that this is using 3 sectors instead of 4, but other than that, it's good. Then there's a video on beta hemolysis, alpha hemolysis, and gamma hemolysis. Watch that. Excuse me, I've got a tickle in my throat. I have allergies and we had another storm come through, so that's my problem. McConkey's agar, we have a video on that. You only need to watch that to 3 minutes and 50 seconds. So it's a longer movie, you don't need to watch beyond that. There's mannitol salt agar. There's a little video to watch on that. And then there's practice preparing a street plate. And this is a virtual lab. Oops, I don't think we're going to do this. Let me check and see if, oh, that's the wrong one. Come on. Let me see if there's a virtual lab in today's assignment. My teacher lab, I haven't updated. This is the lab you guys have. And yep, no virtual lab today. All right, and then work on the questions for the worksheet. When you're done, submit the worksheet for lab 5. Any questions? If not, I'll be here till 8:20 or the last student leaves, and I will be here to answer your questions. So get started on the lab.
I have an irrelevant to the lab.
You have another question? It's not a lab question?
No, it's not a lab question. I was curious. I was looking for the unknown project, the instructions on it, but I can't seem to find it anywhere.
I just showed it. It's under lab 5. Oh, I don't have that on. Let me turn that on. That's why you can't find it. There, now you should see it. Let me see what else needs to be turned on. All right, so it's lab 5. Thanks for pointing that out. And it's the unknown instruction. You may need to refresh your browser, by the way, to see that, or refresh the page. I should say it that way.
Oh, I see it now. Thank you.
Okay. Yeah, I need to turn on all of these labs. Well, let me stop the video here.